Yeah, complement system.
Yeah,
Yeah.
Bismillahirrahmanirrahim. Today's lecture is complement system. We will define what is first complement system. The blood and the other extracellular fluids, they contain numerous proteins with antimicrobial activity, some of which are produced in response to infection. The most important of these are the components of the complement system. The complement system is a part of the immune system that enhances the complement the abilities of the antibodies in the phagocytic cells to clear the pathogens from organisms. There are about 30 proteins produced by the liver or the complements. Complements are the proteins. So there are 30 proteins in the body which is produced by the liver. These proteins, they remain inactive in the blood or in the body or in the tissues after their productions. So they become activated when they are antibody or are formed and they activate the systems, which is known as complement systems. These proteins which are produced by the liver, there are 30 in number, and they are inactivated, and they can be destroyed at 56 degrees centigrade heated for half an hour. So they can be destroyed if they are heated at 56 degrees centigrade. And, and 56 degrees for half an hour. So the complement system is of some non-specific protein which are factors present in the normal human and animal serum. They are activated by antibody reactions. They have the ability to lyse or damage the cells, mediate a number of biological activities like inflammation, muscle permeability, smooth muscles contractions. Complement is a system of some non-specific proteins that are factors. It does not bind to free antigen or antibodies. It does not bind to free antigen antibodies. They bind only with antibodies which are already combined with antigen. But it binds to the Fc fragments of the immunoglobulins. We have different type of immunoglobulins, that is the antibodies, IgM antibodies or IgM immunoglobulin, IgG, IgE, IgA, they are all antibodies of immunoglobulin which take parts in these complement systems. Binding results in activation classical complement pathway. Complement does not bind with all the classes of immunoglobulin. They bind only with IgM and IgG antibodies. These are the two antibodies or the immunoglobulins which take part in the complement system activation. This is a diagram of antibodies, IgG antibodies. You can see it is already labeled and you can read it down. This is the Fc region and this is the Fc regions and these are the Fab regions where they combine together. The Fc region, which is also known as fragment crystallizable regions, is a tall region of an antibody that interacts with the cell surface receptors called Fc receptors and some proteins of the complement system. This region allows antibodies to activate the immune system, for example, through binding to the Fc receptors. the two fragments antigen binding domains FAB and the FC with the two Fab linked to the FC by a hinge region. This is the hinge region. This is the hinge region. So they combine together. The FC combined with the Fab region with the help of these hinges, and allows the Fab a large degree of conformation, flexibility relative to the FC fragments. It is activated, as already told you, by binding of an antigen and antibody. And they are formed a series of reactions. Then a series of reactions take place, and these reactions are known as cascades. These initial components act as enzymes of the following steps. These enzymes cleave the next members of the cascade into larger fragments and the smaller. The larger fragments join the cascade by the smaller fragment, mostly biological effect, and the biological effects, and there may be inflammation, vascular permeabilities, smooth muscle contraction, chemotaxis, detoxification. The first to identify classical pathway, specific active immunity. Most common is the alternate pathway, which is non-specific innate immunity reaction. The complement system, the defense system consisting of over 30 proteins, as I already told you, produced by the liver and found in the circulating blood serums. These proteins are not immunoglobulins, and their concentrations in the serums do not increase after immunization. The complete activation leads to the lysis of the cell and to the generation of many powerful biological active substances. Complement systems work as a cascade systems. Cascade is one reaction triggers another reactions which become trigger another and so on. One after another, the reactions are taking place in the cascade system. And these type of systems can grow exponentially very fast. The complement proteins are often designated by an upper letter C and are inactive until they are split into products like C1. So when the products are split, they become active. The active produced products are usually designated with a lower case A or B, like C1A, for example, and C1B. They will be split like this. The complement system, four important functions, the complement system. They lysis the cell, opsonization, activation of the inflammatory response, and clearance of the immune complexes. Here there are four components. The lysis is taking place here. Destruction of the cell is taking place. Opsonization is here. Then the degranulation take place. And finally, the antigen antibody formation complex, which activates the system. This is the one lysis, opsonization. As you know, opsonization is the process of recognizing and targeting the invading particles for phagocytosis. Means opsonization is the process of recognizing and targeting the invading particle for exozyme, for phagocytosis. Phagocytosis is also a process in which the macrophage, which is a phagocytic cell, it tries to engulf the bacteria or internalize the bacteria to go into the phagocytic cell macrophages, and there it is internalized and kills the macrophage. We have a C3b is an opsonate. That means it binds to both the bacteria and the phagocytic cell, that is macrophages, which helps in phagocytosis. C3b is a component which is formed during the cascades, which is also known as opsonate. C3P is also known as opsonate. That means what is called opsonate because it binds to both the bacteria and the phagocytic cells. So this binds to the bacteria and the phagocytic cells, and which help in phagocytosis. In complement system, there are three type of complement systems. The activation pathway. One is the classical pathway. Number one is the classical pathway. The number two is the alternative pathway, and the number last one three is the menin binding lectin pathway or MBL pathway. We call it MBL pathway, menin binding lectin pathway. So we have three. The classical pathway, in which the antibody complexes are formed. The alternate pathway, aggravated immunology, immunoglobulins and microbial products. And finally, third one is the menin binding lectin pathway or MBL pathway, in which the microbial products take place. So we'll go one by one. The classical pathway, the alternate pathway, and the lectin pathway or the MBL pathway. The classical pathway, the activator is the antigen antibody complexes formation of the antibody involve IgG or IgM only. Only these two. If IgM is involved, it has only one IgM antibody will be involved in this classical pathway. In case of IgG body is involved, then we require two IgG antibodies because the molecule of the IgG is smaller than the IgM. That's why we need two. Whenever there is a classical pathway reactions take place, there we need two IgG antibodies due to the smaller molecule. So we take two, we have to do IgG antibodies, and if IgM is there, then only one is sufficient for the activation of this classical pathway. The activation is an orderly fashion of 9 major proteins component. The major protein which is produced by the liver is denoted as C and is further divided into C1, C2, C3, C4, C5 up to 6, C9. The product activation of the enzyme that catalyzes the subsequent steps. This is IgG antibody. These are IgG. There are two IgG antibodies. These one is a Y shape with IgG antibody. 1 and 2 IgG, some smaller molecule, and this is the binding of the protein. It binds like this to the IgG antibodies. Here another the classical it will bind like this. This is C1 protein, complement protein, and this is microbe, the cell wall of the microbe is containing antigen on the surface of it. So this is antigens. The antibody, these are IgG antibody. They come in contact with these antigen. They bind together with antigen, and the reaction will take place. The activation of the classical pathway will take place. In the activation of classical pathway depends upon the antibody. The pathway is activated when antibody binds to a specific antigen on the surface of the pathogen. Just now I show you in the diagram. Antigen-antibody complexes are formed in the classical pathway. So, three things to be remembered. The activation of this pathway depends upon the antibodies. Second is the pathway is activated when antibody binds to a specific antigen. So the antigen-antibody complexes are formed. And this is the activation of the classical pathway will take place. Diagrammatically, I can explain here the whole classical pathway. It starts from here. This is a microbe, and this is the surface of the microbe, which contain antigen, and these are the IgG antibodies. They bind to the antigen side of the microbe. When they bind to the antibody bind to antigens, the activation of the this take place. The activation of the classical pathway they take place. So when the activation of the pathway, the C, as I told you, there are C1, C2, C3, up to C9 components of the protein. So here, when the activation takes place, there is C2 and the C4, they are cleavage into, the C2 is cleavage into C2A and C2B. Similarly, this C4 will cleavage into C4A and C4B. Here the A is anaphylotoxins. So anaphylotoxin usually does not take part in the cascade, but the molecule of this C2A is bigger than the C2B. So here the C2A will take place in this cycle, in the complement system, and from here C4 is cleavage into C4B and C4A. So here C4A, which anaphylotoxin will go away, will be refused away, it will not take part here, this A. So we are left with C4B and C2A here. C4A and C4B and C2A are left, and this combination of C4B and 2A is known as C3 convertase enzyme. Is known as, or the formation of an enzyme called C3 convertase. This C3 convertase will convertase or cleavage into two portions. C3 will be cleavage into two portions, C3A and C3B. Here, the C3A the anaphylotoxin will be fused away, goes away from the cascade. C3B is left, which combines with this C3 convertase. So we will get C4B, 2A and C3B. We get this another enzyme which is known as C5 convertase enzyme. Understand? So after forming C3 convertase, we get C5 convertase enzyme, and this C5 convertase enzyme will cleavage or converted the C5 complements into two components or fragments, which is C5A and C5B. This C5 is converted into two fragments or components with the help of this enzyme, C5 convertase enzyme, C5A and C5B. So, as you know, C5A anaphylotoxin diffused away, is not taking part in the complement system. The C5B will be here. So this C5B, now they will combine with another components of the system, C6 and C7. This C5 will combine with this 6 and 7, and there is a polymerization of the abundant productions of these proteins are here, and hence this 7 will combine with the 8 and ultimately 9. So the polymerization is also taking place. This is the surface of the cell. So after all combining these components, C5 combined with C6, 7, and 7 combined with 8, and 8 combined with the 9. This is 9. The polymerization is taking place, and this is the surface of the cell where the pouring of this will take place. Pouring of the surface of the cell membrane is taking place. When the pouring of the cell membrane is taking place, everything will be come out from the cell, and the cell will lysis. So this is the death of the cell, and hence this way the complement system works away and kill the pathogens. I hope you remember all this figure. Now, this was the complement system activation, and there. This is all which I already explained you. Okay. C3B is a opsonate. We'll go for another pathway. That is already I explained you. Membrane attack is formed. MAC is a membrane attack complex. It is formed in the end when C6, 7, 8, and 9 they combine together and they form a complex which is known as MAC, MAC, membrane attack complex. And this membrane attack complex, they go and they form the pores in the membrane of the cell. In this way, everything, the electrolytes and all the material inside the cell comes out of the cell, and in this way, the lysis of the cell takes place by the formation, after the formation of the membrane attack complex. Now, the alternate pathway. The alternate pathway have 4 components: C3, factor B, factor D, and properties. Triggering substances may be pathogens or the non-pathogens. Bacterial cell wall components, fungi, viruses, parasites, immune complexes, RBCs, or the polymers. These are the triggering. It triggers the alternate pathway. An alternate pathway have 4 components: C3, factor B, factor D, and property protein. The alternate pathway is one of 3 complement pathway that opsonizes and kills the pathogen. The pathway is triggered when the C3b protein directly binds to the microbes. As I already told you, C3b is the opsonate. It will combine with the microbes also, and it will also combine with the macrophage. Second one is the antibody independent complement pathway. In this, we have factor B. Factor B is activator fluid phase, C3, and forming the C3 converted enzymes, which is C3B and BB. In factor D, what we have factor B in this alternate pathway, it leaves the factor B into BB and BA. Protein is a P which helps in attacking the C3B molecule towards the cell membrane or the pathogen. Factor D splits the factor B into BB and BA. And we are left with a protein, which is protein, which helps in attacking the C3b molecules towards cell membrane or the pathogen. So these are the components of the alternate pathway. The activation of the alternate pathway does not require antibody or immune complexes and is activated by any foreign surfaces like fungal, bacterial polysaccharides, LPS, lipopolysaccharides of the cell, particles, and biometrical surfaces. In this diagrammatic, it is showing the alternate pathway, how it happens. The C3 component is hydrolyzed spontaneously, and C3 fragment attracts to the foreign surfaces. So the factor B binds to C3D, exposed site attracted by the factor D, and cleavage of generates and forming the C3b-BB, which is C3 converted enzymes. C3 converted enzyme is C3b and BB. This is the one is the C3 converted. This is C3 converted enzyme. C3b, BB is a converted enzyme. Binding on the property stabilizes the converted. The protein which we have, which is known as property, which binding will take place of this protein and this binding will stabilize the converted enzyme. The enzymes which are formed, they can easily convert to the next step. The convertase generate C3b. Some binds to C3 convertase, activating C5 convertase enzymes. This C3 convertase enzyme, C3-5 convertase enzyme is here. It is forming here, and hence as I already told you, the alternate pathway initiate take place when through the binding of the C3b onto foreign surfaces. Spontaneous hydrolysis of the internal cluster group of C3 occur in the fluid phase to generate C3 water. This hydrolysis C3 can bind and activate the factor B and cleave the other C3 molecule into C3A and C3B. The factor D cleaves the factor B into BA and BB. The alternate C3 convertase enzyme is formed, which is known as C3B-BB. This is the C3 convertase enzymes. This is the whole structure of the alternate pathway, how it's happening. Here, see the C3 convertase enzyme is formed, which is C3B-BB, and this is the protein which binds with this and stabilizes the enzymes. And then ultimately is the formation of C5 convertase enzymes. C5 convertase enzyme is formed. This C5 convertase enzyme converts the C5 into C5A and C5B. A will anaphylotoxin goes away, and this C5 will combine with the C6, 7, 8, and 9 ultimately, and hence MAC is formed. Membrane attack complex is formed. This is membrane attack complex. Membrane attack complex is formed, which invades the membrane of the cell, and it causes pores on the surface of the membrane of the cell, and hence this way it lyses the cell and kills the cell. That's what it is. This is all about the alternate pathway. It starts from here, as I told you, goes back. This is C3, is spontaneous hydrolysis taking place. C3 water is formed. This C3 is a fluid phase. It is also known as a fluid phase, in which C3 convertase enzymes are formed here. And just there is a factor B and D, which is cleaved into B and BB. Factor B is cleaved by the factor D into BB and BA. Then C3 convertase is converted into C3B and C3A after forming the C3 convertase enzymes. It is broken up into two fragments, C3A and C3B. And hence the properties is bind to the C3 convertase enzyme to form the stability of the enzyme, and ultimately C5 enzyme, which is C3B, BB, and 3B. This is a C5 convertase enzyme, which converts the C5 fragments into two portions, C5A and C5B. A goes away and anaphylotoxins won't take part in these cascades. This C5 is left, which combines the 6, 7, 8, and 9 and forms the complex which is known as MAC complex, membrane attack complex, which ultimately is the surface of the cell of the pathogens. It pores the membrane of these pathogens, and hence the lysis or the death of the cell will take place. This was the alternate. This was the alternate pathway. Now we have another last one is the MBL lectin pathway or mannose binding lectin pathway is the third one. pathway, manose, binding, lectin pathway for MBA. It does not depend on the antibody for activation. Originate with the host protein, which is the MBL protein, binding with the microbial surface. Okay. The MBL pathway, while initiating of the lectin pathway, takes place through manose binding lectin. Lectin pathway is complement pathway that activates the complement system by binding of the MBL to the sugar or the carbohydrate which is present on the bacterial surface. The manose binding lectin pathway acute phase protein binds to mannose residue and to certain other sugar on many pathogens. MBL like C1Q is a 2 to 6 hydrogen molecule that forms a complex with two protease enzymes, which is known as protease zymogens or MASP1 and MASP2. These are two enzymes, two protease enzymes. Menin binding lectin, which is MBL, associated with the serine protease 1. When the MBL complex binds to the pathogen surface, MASP2 is activated to cleave C4 and C2. C2 is cleaved into C2A and C2B, while the C4 is cleavage into C4A and C4B. The C3 converting enzyme is formed from C2A bound to C4B. This is the C3 converting enzymes. C4B and 2A is formed, which is the C3 converting enzymes. MBL pathway is important in innate host defensive mechanism in the early child. The lectin is a protein that binds to the carbohydrate. Mannose binding lectin, it binds to the mannose or many bacterial cells. MBL is produced by liver in acute phase and inflammatory reaction. Once MBL binds to the target cell, two serine protease enzymes, MASP1 and 2, they bind and act like a C1 as it happens in classical pathway. And the MBL pathway is initiated by the pathogen membrane containing carbohydrate like mannose or sugar. In this, we have the following components in this MBL pathway. We have number one component, MBL, which is mannose binding lectin, MASP1 and MASP2, which are two enzymes. But in all these three components, they join together and form a complex from the pathogen membrane during lectin pathway. This complex is known as mannose complex, and it's resembled like C1 complex of the classical pathway. And this mannose complex binds to the pathogen surface. MASP2 is activated to cleavage the C4 and C2 components. Well, diagrammatically, I can explain you this MBL pathway. This is microbes. This is the surface of the microbes where carbohydrate, mannose or sugar is here present on the surface of the microbes. This C1 like components which comes and attached to the surface of the this microbe where carbohydrate or sugar or mannose is present here. The MASP2 is activated here and cleavage C2 and C4. So C4 component and C3 components and C4 2 and 4. This is 2 and 4 component. C2 is cleavage into C2A and C2B. C2A and C2B. A goes away of C4A. The C4 is converted into C4A and C4B. As, so they combine together and form the C3 converted enzyme, which is C4B. This is C4B, this is C4B, and 2A of this C2 component, C2A. These are the two components they form together an enzyme which is known as C3 convertase. This C3 convertase will convert C3 into two components, C3A and C3B. The CA goes away, the anaphylotoxins here, and the C3 is left, which forms another enzyme, which is known as, which is C35 convertase enzyme, which is C4B, 2A, and 3B. This is C5 convertase enzyme. This is C3 convertase enzyme, this is C5. The C3 convertase cleavage C3 into two portions. Similarly, the C5 convertase enzyme will cleavage C5 component into two components, C5A and B. The C5A is anaphylotoxin, goes away, not taking part in the cascade there. Only C5B is left, which later on combines with the 6, 7, and 8, and ultimately polymerization of C9 take place, and all together combine together and form a complex, which is known as MAC, membrane attack complex, and hence they pore the surface of the, this is the surface, this is the surface of the pathogens, and pouring of the pathogen membrane cells take place, which kills or lyses the cell integrity, and hence the pathogen is killed to clear away from the body. Like C1 has happened in the complement system, which already I explained you all these things. Okay. These all already I explained you. The last one was the manase MBL lectin pathway. As I told you, we have three classical pathway. We have the classical pathway in which anti-antibody complexes take place. MBL pathway in which mannose binding lectin binds mannose on the surface of the pathogens. And they have two in line, MASP1 and MASP2. And the second one was the alternate pathway in which the pathogen surfaces take place. C3B, factor D, factor B take place in these enzymes and forms the C3 convertase enzyme. Ultimately, and MAC is formed, which is known as membrane attack. If we have a deficiency of the complements, certain complement, we can have some diseases. In case of complement C3, the deficiency C3 and factor B, a severe bacterial infection can take place. If there is a deficiency of C3B, C6 or 8, even a severe infection can take place. Similarly, C4, 1, 2, and 2 deficiency can cause SLE and glomerulonephritis and polymer. A kidney problem can take place. The C1 inhibitor can inhibit angioedema. The biological effects where they may be of the complement may be oxidization. C3B is oxidizing molecule. It means that it binds both to the bacteria and the phagocytic cell macrophages, which hence increases the process of phagocytosis. So C3B is also known as opsonine. In case of chemotaxis process, C5A and C5-6 complex, they attract the neutrophil, and C5A enhances the adhesiveness of neutrophils to the epithelium. The anaphylotoxins which have A, C3A, C4A, C5A, they stand for anaphylotoxin. A stands for anaphylotoxin. For the degranulation of the mast cells, bind directly to the smooth muscles of the bronchioles, and hence the spasm of the bronchiole can take place. This is oxidation and the phagocytosis we already explained here. Fourth one is the cytolysis or the MAC formation, disrupt the membrane on entry of the water and electron into the cell. Enhancement of the antibody production. Binding of C3B to its receptors on the surface of the activated B cells enhance the antibody production. The role of complement in disease. Complement systems play a critical role the inflammation and the defense against some bacterial infection. Complement may also be activated during reactions against the incompatible blood transfusion and during the damaging immune system responses that accompany the autoimmune disease. Deficiency of the individual complement, component, or inhibitor or systems can lead to a variety of diseases. Another one is the role is the complement system helps antibodies and phagocytic cells to clear the pathogen from the organism. The complement system consists of a number of small proteins produced by acute phase reaction in the liver during production. The complement systems might play a role in diseases with immune component and those of the central nervous system. Complement protein deficiency also is a form of primary immunodeficiency. Thank you very much. Yes, and the complement system. Uh, huh? Ali, Ali को बुलाना ना? क्योंकि पूरा ने स्टॉप शेयरिंग कर दिया आगे बॉडी की बड़ी आपने
स्टॉप शेयरिंग
हाँ वो किया एंड होगी ना
एंड यहाँ से